inviting me here today. So I'm visiting from the Department of Anthropology at the University of San Diego. And I'm going to speak today about some of our earliest human ancestors. And specifically, I'm interested in the point that our hominin ancestors began eating meat. This is a big change in behavior, and it's correlated with the advent of tool use, and it's also correlated with an increase in brain size. I'm an archaeologist, and so I'm going to talk about some of the archaeological evidence that documents how our ancestors acquired meat, how they competed with other carnivores to get it, and ultimately how this change in diet affected the evolution of human social behavior. So just to give you an outline of what I'll be talking about, I'll start by giving some background about the evolutionary context of meat eating. Meat eating is so important because it's probably what triggered some of the major behavioral changes that took place in our hominin ancestors. Particularly, it's probably responsible for the evolution of cooperative social behavior, and this is a hallmark of modern human behavior, so this is why I'm focusing on meat eating here. I'm going to tell you about some of my own research on the fossil evidence for meat eating in East Africa and how experimental archaeology can be used to interpret this evidence. And I'll finish by talking about some of the implications for understanding the evolution of human behavior, and then also a bit about my ongoing field research. So to begin with, who are our fossil hominin ancestors? We've all probably seen some kind of scenario like this cartoon showing a linear progression from an ape through several phases of development, ending with a modern big-brained technology using human. But this isn't quite how it worked. The pattern that human evolution has followed looks something more like this, like a branching tree. And the base of the evolutionary tree extends back past 6 million years ago, when we share our last common ancestor with the chimpanzees. All the fossil species on this tree that are collectively referred to as hominins, they include several groups. So we have the Australopithecine group, Paranthropus right here, and then our own branch on the tree, the genus Homo. And we're the only living members of the genus Homo. Now the hominin evolutionary tree is constantly being refined, and pieces are being filled in as we discover new fossils or we learn more about fossils that we've already discovered. But one particularly interesting part of the tree is right here at about 2.5 million years ago. And this is the origin of the genus Homo. The earliest species belonging to the genus Homo was discovered by Lewis and Mary Leakey at Olduvai Gorge in Tanzania in 1960. They named it Homo habilis, or handy man, because it was thought to be the first to make stone tools. And in their 1964 paper announcing the species, they argued that Homo habilis fit 3 key criteria for belonging to the genus Homo. It had upright posture, it had a bipedal gait, as evidenced by these foot bones that they found at Olduvai Gorge. And it also had the dexterity in its hands to fashion primitive stone tools. Now, paleoanthropologists were reluctant to accept the earliest finds by the Leakeys as a brand new species because they were pretty fragmentary. But later on, some more complete specimens were discovered at a site called Kubifora in Kenya. This is one of them, this skull, the 1813 skull. It's actually one of the most complete specimens of Homo habilis that we have. So Homo habilis today is generally accepted to be ancestral to later species in the genus Homo, including Homo erectus. However, there's some variation in the fossils that date to this period, so Homo habilis might actually represent more than one species. So for this reason, researchers sometimes collectively refer to this group of fossils from this time period as early Homo. So this is the famous Lucy skeleton here from Ethiopia, Australopithecus afarensis, and it's likely the ancestral species to genus Homo. If we fast forward a million years later, we find something very different. This is Homo erectus. It's the Turkana boy skeleton from near Lake Turkana in Kenya. Now, during this transition from Australopithecus to Homo erectus a million years later, there were some important anatomical changes that took place. And these include a shift to dedicated bipedalism. So in other words, members of the genus Homo began walking on 2 feet and only 2 feet. Brain size increased, and body proportions also became more modern, which you can see just by comparing Lucy to the Turkana boy here. And here we have a time scale to put this in perspective. The origin of bipedalism dates back to around 6 million years ago, but it's not until about 2.5 million years ago with the origin of the genus Homo that we see this shift to dedicated bipedalism. And although the Australopithecines and the earlier hominins were bipedal, they retained some more ape-like characteristics, including long arms and more ape-like body proportions, which you can see comparing this Australopith skeleton to a chimpanzee and to a human. And these things like long arms and these ape-like body proportions, these are adaptations for climbing in the trees. Now, it was early members of the genus Homo that were the first to shift to dedicated bipedalism. So the question is, why walk just strictly on 2 legs? And the reason is thought to be climatic change. So beginning around 5 million years ago, sub-Saharan Africa underwent this cooling and drying phase. And this is what led to the spread of the savanna grasslands that we see in East Africa today. Australopithecines lived in forest habitats and probably relied on eating woodland plant foods that they found in these habitats. And so for the Australopithecines, this change in climate would have meant that their food resources would have become more scarce and would have required that they range further between food patches every day to find food and expend more energy in their search for food. Now, it turns out that walking bipedally actually uses less energy than walking quadrupedally. So this switch to bipedalism probably aided hominins in finding food during this climate shift because they could more easily move between these food patches that were spread further and further apart. Bipedalism isn't the only major transition during this period, though. Brain size also increased. So where Australopithecus afarensis had an average brain size of around 450 cubic centimeters, the brain size of Homo erectus had increased to around 900 cubic centimeters. For comparison, the human average is about 1350. And it turns out that brains are actually extremely energetically costly organs to run. They account for between 16 to 20% of our daily energy budget. And if we looked at the amount of energy usage by unit weight, the brain uses roughly 16 times as much energy as muscle tissue does. So it's a very costly tissue to maintain. And for humans, we use a very large share of our daily energy budget just to feed our brains. So for early hominins, this would have meant that an increase in brain size would require that they adopt a diet with enough calories and enough nutrients in it to meet these new energy demands. So the Australopithecines and the earlier hominins probably lived in forest environments that may have looked something like this. This is a photo that I took in western Uganda last summer, and there are plenty of plant foods for a primate to eat in an environment like this. Beginning around 5 million years ago, the spread of the grasslands in East Africa would have limited the amount and the variety of edible plant foods available to hominins. And fossil evidence suggests that different lineages of hominins coped with this environmental change in different ways. So, for example, the Paranthropus lineage was a dead-end branch of the human family tree that lived alongside members of the early genus Homo. And Paranthropus coped with this change anatomically by evolving specializations like huge teeth and huge jaws that allowed them to subsist on the more widely available but probably more difficult to chew foods like seeds or plant roots or more fibrous plant material. Early genus Homo took kind of a different path and began to exploit some of the new resources that became available in these grassland environments. Now, as it turns out, the spread of the grasslands also led to an increase in grazing mammals like antelope. And animal tissue is very dense in calories and nutrients, far denser than most plant foods. And so hominins who were capable of exploiting these antelope on the African savanna would have had an advantage. Evidence suggests this is the strategy followed by early Homo. This is Homo habilis pictured here. Homo habilis had smaller teeth and more delicate jaws that were not adapted for chewing tough plant material. But one thing that Homo habilis did have was tools. The emergence of the genus Homo is associated with the earliest stone tool industry. It's called the Oldowan tool industry after Olduvai Gorge, where these first tools were discovered. This is one of the earliest Oldowan tools. It's from a site called Gona in Ethiopia, and it dates to 2.6 million years ago. Also associated with Oldowan stone tools at several archaeological sites are fossil antelope bones that have butchery marks on them. So this indicates that hominins were butchering these animals, and presumably they're butchering them because they're eating them. So these changes in the brain size and the body size of early Homo compared with their Australopithecine ancestors suggest an increased energy requirement, especially by the time we get to Homo erectus. Increased energy expenditure would have required a higher quality diet, and meat may have been an important component of this. So with the early genus Homo, we're seeing a significant shift in diet to include more nutrient-rich foods like meat, and these are foods that are being acquired with tools. Now, this initial increase in brain size and then at the same time, the shift to a higher quality diet would have set the ball rolling for additional behavioral evolution, with bigger brains producing more complex foraging behaviors that could improve diet, and then this in turn would have spurred additional brain evolution. So eating a high quality diet is probably closely linked with the increase in brain size throughout human evolution. Now, we know that early hominins were eating meat because there's archaeological evidence for it at several sites in East Africa where we find these really dense concentrations of bones and stone tools. And these sites indicate that hominins were returning to the same places on the landscape to process animal carcasses. Now, just because hominins began including larger quantities of meat into their diet doesn't mean that they turned into carnivores. What we're talking about here is a more gradual shift from an ape-like diet that included woodland plant foods to a diet that included more nutrient-rich foods found in savannas, and these are foods that are harder to acquire. Meat was one of these foods, and meat is the one that leaves a mark in the archaeological record. So bones survive, and plants usually don't in the archaeological record, so it's easier to find evidence for meat eating. Plant foods probably were a very important part of the hominin diet, though, just as they are in modern human hunter-gatherer diets. Some of the plant foods that, and other foods that hominins probably ate included wild tubers, just as hunter-gatherers do today in Africa. This is a Kunsan man in the Kalahari of South Africa harvesting a wild tuber. Tubers are a good source of carbohydrates. Tubers are pretty common in grassland environments, and so hominins probably would have exploited that resource. They may have also eaten a number of other gathered foods like berries. These are Kongolobe berries, which are a staple in the diet of the Hadza hunter-gatherers in Tanzania. The Hadza also eat honeycomb. Honeycomb is high in calories, and so if you can get at it, it's a good resource. So hominins probably were eating a lot of plant foods. Plants don't survive in the archaeological record for us to find evidence for. One way that we can get at plant eating, though, is to look at the use wear on stone tools. So using stone tools for different types of functions can leave microscopic wear patterns on the cutting edge of the stone tool, like right here. To try to figure out what hominins were doing with these stone tools, some of my colleagues and I conducted a study where we made our own stone tools, and we used them to process different types of foods, and then we compared the wear patterns on our tools to see if they resembled the wear patterns on on tools from Oldowan sites. These are some of my colleagues cutting grass with some stone tools. These are some of the tools that we made in our study. And when we analyzed the use wear on tools from archaeological assemblages, what we found was it looks like hominins were using Oldowan tools for a whole variety of purposes. So they were cutting wood, they were cutting grasses or reeds, they were butchering animals or cutting meat, and they were peeling tubers. Why would hominins be cutting wood with stone tools? They may have been sharpening digging sticks to help them get tubers out of the ground. So hominins were eating plants as well and using tools for this whole variety of purposes. We just don't see as much evidence for plant eating in the archaeological record. So this is just to keep in mind that hominins were eating plants, but I'm focused on meat here because the shift to including more meat in the diet probably had a major effect on hominin social behavior. Now, while we do know that hominins were butchering animals and eating meat, there are some things that we don't know, and these are things that have important implications for understanding hominin behavior. First of all, we're not sure how these animals were acquired, whether it was through hunting or scavenging. Scavenging is a much less risky activity, and hunting is more dangerous. So if hominins were hunting, this means they were probably cooperating as a group. We also don't know how much meat they were getting. If they were getting scraps of meat, that would probably not be socially important because it's hard to share a scrap of meat with someone else in your group. But if they were getting big packets of meat, that would make it a shareable resource. And then we also don't know how they interacted with other carnivores in their environment. So how did they compete with carnivores to access meat? Speaking of other carnivores, the African savanna was a much more dangerous place 2 million years ago than it is today. There were more species of carnivores, and hominins had not yet reached modern human body size. They were between 3 and 4 feet tall. So the savanna would have been a pretty dangerous place 2 million years ago. These are some of the modern large carnivores of East Africa. We have the big cats, lions, leopards, and cheetahs. In East Africa, there are 2 species of hyenas, the spotted hyena, which is the larger species, and the striped hyena. We also have African hunting dogs. Hunting dogs are not, they don't have a large body size, but they do hunt in packs, and so they can be somewhat intimidating. Now, in addition to the modern big cats in Africa, there were 3 species of saber-toothed cats that lived during the early Pleistocene. Dinofelis, Meganterion, which had the largest saber teeth, and then Homotherium, which was the largest body size species. They had a body size larger than modern male lions. And fossil evidence suggests that Homotherium actually specialized on preying on juvenile proboscideans. So there were these 3 big cats, and then also there was a species of giant hyena that's now extinct called Pachycracuta. This in blue here is showing the size of Pachycracuta compared to the size of the modern spotted hyena, which is the largest extant hyena. So these carnivores would have posed a significant threat to hominins. And when hominins began to include meat in their diets, they would have had to evolve strategies to be able to compete with these carnivores. And we know that hominins themselves sometimes ended up on the menu because we found fossils of hominins with tooth marks on them. So we know hominins were eating meat starting around 2 million years ago. They were running around bipedally on the savanna using their big brains and their stone tools to figure out how to get meat, and they were competing with carnivores over access to meat. So this is where my research questions come in. I want to know exactly how hominins obtained meat and what this may have meant for the evolution of more modern human behavior. So back to my research questions. I want to know, did hominins have early access to carcasses? And if they did they acquire these carcasses through hunting? Or did they gain later access to carcasses by scavenging from other carnivore kills? Or maybe it was some kind of combination of these strategies. I want to know how much meat they were getting. Was it a very important component of their diet? And I want to know how hominins competed with carnivores in their environment. So overall, how did they fit into their broader ecology? To answer these questions, I've examined patterns of damage to fossil bones, so looking at tooth mark damage and cut mark damage on fossils from 2 Oldowan sites. And to help me interpret these damage patterns, I've conducted experiments with modern carnivores to model how they actually damage bone. And in this analysis, I've examined the frequency of tooth marks in my models, and I compare them to the fossil assemblages. And then one of the methods that I've applied is to use GIS to visualize patterns of bone damage, and I'll tell you a little bit about that in a moment. So when we're thinking about scavenging, this activity could take a few different forms that are actually behaviorally quite different. So one version of the scavenging hypothesis views hominins as these more passive scavengers. So they're waiting in the bushes until the lions and the hyenas are totally finished feeding before venturing out to eat whatever scraps are left over. This passive strategy is safest, but it potentially wouldn't yield enough meat to make it a very dependable strategy. On the other hand, a more active, aggressive confrontational strategy where hominins actively scared carnivores off of their kill right after the kill was made, this could have yielded potentially much larger quantities of meat. This would be a more dangerous strategy, but one that potentially would have had a pretty high payoff. So how would hominins have done this? They would have had to throw things and make a lot of noise, something like what's going on in this image here. But importantly, single individuals probably would not be doing this. This is the kind of behavior that would have been a cooperative group endeavor. So my study focused on bone surface damage on fossils because bones can give us all kinds of information about what hominins and what carnivores were doing in the past. Carnivores modify bones in several ways that are recognizable and in ways that give us information about what they were doing. So they might transport bones away from a kill site. Hyenas are famous for doing this. They might rip a limb off of a carcass and transport it back to their den. Carnivores might differentially consume or destroy different portions of bones. So for example, hyenas tend to destroy the ends of bones because that's where the grease is, so they might crunch off the end of a bone. But probably the most distinctive signature that carnivores leave on bones are tooth marks, like these. These are modern tooth marks in this photo here, but these types of marks actually survive on bone and are visible on fossils as well. One of the main things that affects scavenging opportunities that might have been available to a hominin is the adaptation of the initial carcass consumer. So hyenas, for example, have these incredibly strong teeth and jaws that are specialized for bone crunching, and so they're more able to fully consume a carcass. On the other hand, lions are flesh specialists, and they typically don't break bones or consume the marrow. They're really just focused on the flesh. Saber-toothed cats were probably pretty similar to lions in this adaptation because their large canines would have been at risk of breaking if they really tried to crunch bone. These canines are actually relatively fragile. So saber-toothed cats were also likely flesh specialists. So carnivores leave tooth marks on bone. Humans also leave distinctive marks on bone when they butcher them. So these are what cut marks made by humans look like. These kind of linear striated marks on bone. These are bones from a modern collection of wildebeest that were butchered using stone tools. And we find these types of cut marks on fossils as well. When humans process carcasses thoroughly, they also often use a hammerstone to break open the limb bones to access the marrow. And this activity leaves a distinctive notch feature along the fractured edge of the bone, adjacent to where the point of impact was. So here. And then if you look at the inside of the bone, so this is the same bone looking at it from the outside and looking at it from the inside. If you look at the inside of the bone, there's often also this flake that's taken off right along the edge of the notch. So these are features that are evidence of or distinctive of hammerstone percussion, or really of active dynamic loading. So you're hitting something in this dynamic way and it's breaking in this pattern, versus the static loading that a carnivore jaw would produce, so pushing down in a more static type of motion. So this is typical of hammerstone damage to bones. So I've analyzed the fossil assemblages from 2 different Oldowan sites in East Africa to look for these types of bone modification. These are sites that date to roughly the same time period when the genus Homo appears in the fossil record. Kandera is located in western Kenya on the shore of Lake Victoria, and FLK Zinj is a site at Olduvai Gorge in Tanzania. FLK Zinj is one of the best studied early hominin sites. Its technical name is FLK level 22 from bed 1 at Olduvai Gorge. And this site was originally excavated in 1959 by Lewis and Mary Leakey after their discovery of let's go back for a second, of this skull. This is Olduvai hominin 5, Paranthropus boisei. When they discovered it, they named it, or they put it into the species Zinjanthropus, so the site is often referred to as Zinj. FLK, by the way, stands for Frida Leakey Karongo. Frida Leakey was Lewis's first wife, and so he named that part of the gorge after her. This is Leakey with the original Zinjanthropus skull. So this site, FLK Zinj, dates to around 1.8 million years ago, and it contains thousands of fossil specimens and Oldowan stone artifacts. Many of the bones show evidence of butchery marks. You can see them on this specimen here. Several years after the Leakeys discovered the Zinj skull, they found the first specimen of Homo habilis in the same level nearby. It was this jaw, the OH7 jaw. This is the type specimen. So it's thought that it was early Homo and not Paranthropus that was the maker of the stone tools and responsible for butchering the animals at the site. Because the collection at Olduvai is so large and because it's really exceptionally preserved, it's been studied a lot, probably more than any other site, for trying to understand evidence of early hominin behavior. But there have been some pretty different interpretations of the evidence by different researchers. The first person to do a systematic examination of the carnivore tooth mark damage on the FLK Zinj fossils was Rob Blumenschein. He was at Rutgers University at the time. And he used modern carnivore feeding experiments to interpret the archaeological evidence. So Blumenschein conducted these experiments mostly with hyenas, and he presented hyenas with bones that had flesh on them to simulate carnivore first access. And then he also presented hyenas with bones that had the flesh removed. So these are his carnivore last experiments. And so this would simulate hominin primary access and carnivore scavenging for something that hominins had already butchered. So this one models what it would look like if hominins were hunting and carnivores were scavenging from them. So he compared these models, and in the carnivore first models, 75% of the limb bone shafts showed tooth marks on them. But when the carnivores were presented with bones that had already been defleshed, they were only tooth marked between 5 and 15% of the time. Now, when Blumenschein compared the FLK Zinj assemblage to these experimental models, he found that approximately 59% of the limb bone shafts were tooth marked. And this doesn't really resemble either model, but it's closer to the carnivore early access model. So this finding seemed to indicate that carnivores had relatively early access to bones at FLK Zinj. But there's also a relatively high frequency of percussion marking on the bones, and this indicates that hominins were breaking the bones open for marrow. So to explain these different lines of evidence, Blumenschein described this 3-stage model where he proposed this scavenging niche for hominins. So he argued that the high frequency of tooth marks on the limb bone shafts indicated this initial access by lions, or primary access by lions. Since lions are flesh specialists, they don't have adaptations for crunching the bone. So if lions had first access, the marrow would have still been intact for the hominins to gain secondary access to. So this next stage he proposed then was secondary access by hominins. And hominins would then have broken open the bones to access the marrow. This would explain the hammerstone damage on the bones. And then he proposed this final stage of scavenging by bone-crunching hyenas to account for the low proportion of limb bone ends in the assemblage. So the assemblage mostly contains the shaft parts of the bones, not the ends. So it might have been hyenas that chewed off the ends. And more recently, other researchers have argued against this three-stage model for two main reasons. A Spanish research group led by Manuel Dominguez-Rodrigo has also analyzed the carnivore tooth marks from FLK zinj. And they've claimed that many of the marks that were originally assumed to be tooth marks were actually produced by fungus growth or some other biochemical processes. Dominguez-Rodrigo's group did these experiments letting fungus grow on modern bone. These are some images from their experiments. And they've argued that the pits that fungus can create on bone actually resemble carnivore tooth marks. These are some photos of marks that I took on the FLK zinj fossils that I actually do think could be the result of biochemical damage. So the tooth mark frequency that Blumenstein originally calculated for FLK zinj might not be as high as he thought because of this type of fungal damage. A second argument against the three-stage model is that the original study didn't incorporate any of the evidence of cut marks. Actually, about 15% of the bones at FLK zinj are cut marked, and this is similar to the frequency that's documented in modern butchery experiments where humans have first access. Also, of the bones that are cut marked, about half of them are meaty portions that occur early on in the consumption sequence. So these are portions that humans tend to go for first because there's a lot of meat on those particular bones. So Dominguez-Rodrigo reinterpreted FLK zinj as a site where hominins had early access to carcasses rather than a site where hominins were scavenging from carnivore kills. So one of the goals of my study was to reanalyze the FLK zinj collection using new methods to see if I could help solve this debate. And one of the major problems with studies trying to interpret hominin meat eating is that we've been basing all of our interpretations on one single site, on FLK zinj. And researchers have really focused on the fossils from Olduvai because they're so well preserved there. But we have no way of knowing if FLK zinj represents a typical pattern of hominin behavior or how variable hominin behavior might have been until we analyze additional sites. Now, the problem is that we just don't have many sites dating to this time period, but the fossils that we're finding at Canberra are giving us a great opportunity to compare with Olduvai. Canberra is in many ways comparable with FLK zinj. The sites are similar in age. Canberra dates to about 2 million years. The sites are similar in size, and they have roughly the same number of fossils and stone tools. There are a few interesting differences, though. The bone assemblage at FLK zinj is made up of mostly large antelope. They're both mainly made up of antelope. But at FLK zinj, they're larger size antelope, like a wildebeest size. The antelope fossils at Canberra are from smaller sized animals, more like gazelle size. And then also the environments have been reconstructed as being a little bit different at these two sites. At FLK zinj, the environment has been reconstructed as a woodland or a grassy woodland. It may have been similar to this type of environment here. This is Nairobi National Park in Kenya. The ancient habitat at Canberra has been reconstructed a little bit differently. It's been reconstructed as a more open grassland, similar to something like this. This is Ruma National Park in Kenya. It's actually not far from Canberra. Canberra is located in western Kenya on the Homa Peninsula of Lake Victoria. Slide's a little bit dark, sorry, but you can see Canberra is right here. This is the Homa Peninsula. This is Homa Mountain. It's an extinct volcano. This is a Google Earth satellite image, and this is literally how we find fossil exposures. We look at the satellite, and the fossil exposures are light colored because these are erosional areas. There's not vegetation growing in these areas, so it's easy to spot fossils. The word Homa, by the way, actually means fever in the local Luo language that's spoken in Western Kenya. And they call it the Fever Peninsula because there are a lot of mosquitoes. So there's actually a lot of malaria there. We never miss a day taking our anti-malarials when we're in the field here. This is what it looks like at Canberra. The area is extremely rich archaeologically. There are a lot of fossils eroding out on the surface. And in fact, there are other fossil deposits all around the peninsula that we've identified in surveys, and we're just starting to explore some of those other areas. But this is the Canberra area. This is what our main excavation looks like. We've had 5 excavations, and this is our largest one, excavation 1, when it's in progress here. The excavation goes pretty slowly and carefully. The sediment is really hard, so it's kind of like digging through cement. Here's some of the artifacts and fossils in the ground. I'll try to point some out here. This is an antelope pelvis right here at the hip socket, an antelope jaw here. There's some other teeth and pieces of fossils from mostly antelope. And then also there are Oldowan stone tools here and here that we find embedded in with these fossils. The fossils preserved at Canberra are mostly antelope and zebra, but there are also pigs, birds, crocodiles. There's one hippo that has cut marks on it. The antelopes are mainly small, like I mentioned, and of those small antelope, about half of them are juvenile. Here's a sample of what some of the limb bones look like. These are all antelope. There are a variety of different types of damage on the Canberra bones that show evidence of both carnivore activities and hominin activities. There are, hammerstone-produced percussion notches here. These are percussion pits. These are carnivore tooth marks here. This one is a carnivore tooth mark overlying a cut mark. So this is a really interesting fossil because this shows that at least in this instance, hominins butchered this bone prior to carnivore gnawing. Here's some of our finds from the Canberra excavation recently. This is an antelope scapula or shoulder bone right here with some Oldowan flakes that are lying right next to it. And these are the kinds of tools that hominins were using to butcher bones. And you can't see it in this photo here, but there actually are cut marks on the scapula. So this is literally the remnants of a hominin dinner right here from 2 million years ago. It's pretty cool. Also, we found some nice Oldowan core tools. This is, what happened to my pointer? This is one of them. This is an elephant tusk. So this gives us some idea of the kinds of animals that hominins were sharing their environment with. This is another one of our recent finds. So in the center of this square here is a cobble that looks like it was used by hominins as an anvil for breaking bones. And all of these little numbered bones here are shattered pieces of antelope limb bones that are lying all around the anvil. And they have percussion marks on them. So this definitely looks like a place where hominins were processing bones for marrow. This is one of those little fragments close up. And if we zoom in, we can see that there's clear evidence of hammerstone percussion marking on this fragment. So these are some of the types of things that we're finding at Canberra. Now, in order to interpret these fossil assemblages, a major part of my research has been to build experimental models of the types of bone damage that carnivores create when they gnaw on bones and that humans create when they butchered bones. This is one of my colleagues butchering a goat limb with a stone tool that we made in one of these experiments. Now, since hominins were competing with carnivores in their environment to access meat, it's really important for us to also understand the signature that carnivores leave in the archaeological record. So I've focused a lot of my research on doing carnivore feeding experiments, and I've conducted experiments with a few different species of carnivores. And my aim was first to increase the sample size of these experimental models that are available. There have been some feeding experiments done by other researchers, like the one that I mentioned by Blumenschein, but they've mostly focused on hyenas and they haven't been very extensive. So my aim was first to see if I could increase the sample size of these experimental models, and then my second aim was to see if I can differentiate patterns of gnawing that different carnivore species produce. So when I'm looking at a bone like this that's been chewed on by a carnivore, can I tell if it was a lion or a hyena that gnawed on it? So I conducted some of my experiments with wolves at the Wolf Conservation Center in upstate New York. The wolves here are being prepared for eventual release into the wild, and so they're kept under more natural conditions than typical zoo animals are. Rather than being fed a diet of zoo chow, they're fed whole carcasses like they would find in the wild. And the carcasses are mostly chickens and road-killed deer that are donated from the New York State Department of Transportation. And it's important for our research that they're getting whole carcasses, because this means that the wolves encounter like fur and tendon and all of the things that come with a whole carcass, and so their gnawing behaviors should be more similar to what wolves do in the wild. Our sample from this study is over 1,300 bones, so there's a lot of roadkill in New York State, and this actually makes this the largest experimental bone collection that's been published so far. I've also recently started a project on lion feeding in Queen Elizabeth Park in Uganda. So my experimental work so far has been mostly with captive carnivores, but I wanted to extend it to look at carnivore behaviors in the wild, which is more difficult to do, but it allows us to observe more realistic feeding behaviors. And I chose Queen Elizabeth Park, which is right here in Uganda on Lake Edward. I chose Queen Elizabeth Park because it has a very high lion density, and so the chances are good for observing lions in action modifying their prey. And also in the early Pleistocene in East Africa, there was a high lion density with these extra species we have that are of saber-toothed cats that are now extinct. So our goal with this study was to locate lion kills and to observe first, what type of prey are they taking? Are they taking large or smaller size antelope? How much flesh do they leave behind for other scavengers? So this is a buffalo carcass right here, and for example, this one was pretty fully consumed, so there isn't much left for scavengers. And then also, I'm interested in what do the tooth marks look like on the prey bones. So this is a buffalo foot bone with some pretty large tooth marks on it. So in order to do this, we walk or drive in 1 kilometer straight line transects across the park to document any kills that we find along the way. And this project is still underway, but so far we've documented 100 lion kills. And this is our team from last summer here. So to analyze the bone damage in my experimental collections and compare them to the fossil collections, I've applied a relatively new method using GIS. Now, if you're familiar with GIS, you know it's a mapping software. But rather than mapping landscapes, what I'm essentially doing is making small-scale maps of bones. So the bone is kind of like my landscape here. I've used GIS to record the location of different types of bone damage on each element. So these include tooth pits, tooth scores, cut marks, and percussion notches. So each tooth pit, for example, gets plotted onto a template like this. And I can then create a composite record of the distribution of tooth pits for each bone. So the example that I'm showing you here is a composite plot of tooth pits on the left femora, all of the left femora in my wolf experiments. And then I can also do the same thing for cut marks. So this is the cut mark distribution on an experimentally butchered bone collection. So these are the areas that humans tend to cut mark the femur. So these are the kinds of data that I'm recording. And then in my analysis, I've used some of the spatial analyst tools in ArcGIS, not just for displaying the location of bone damage, but to try to quantify it in a more precise way. And the cluster analysis is a really useful way to quantify how modifications are distributed across bones. So the cluster analysis takes all the points that I have mapped onto my template here, and it measures the distance between them, between each point, to identify significantly dense concentrations of tooth pits. So on this graphic, the shaded areas indicate the highest concentrations of tooth pits, and the red is the highest. Okay, so that's a bit about the method. Onto the results. These are some bones from my experimental studies, and they show the range of damage that carnivores produce on bones. These are some of the hind limb bones that were gnawed on by lions. These are hind limb bones that have been gnawed on by wolves in my experiments. And you can see that the wolves tend to destroy the ends of the bones more often than the lions do. And in fact, they kind of chew them right off, especially at the knee joint, the end of the femur and the top of the tibia here. The lions tooth mark these areas, but they're less likely to chew off these areas completely. So using these experimental bone collections, I've built GIS models that show composite images of where carnivores typically tooth mark bones. So these are some of the different limb bones in my analysis showing where wolves tend to leave tooth marks. So in my study, I've used these experimental models of carnivore damage to interpret the fossil bone damage from FLK zinj and from Canberra. These are some examples of carnivore damage to bones at FLK zinj. So you can see there's a tooth mark here. This is a tooth mark. This is a tooth mark here. These are some examples of hominin damage on fossils from FLK zinj. This is a percussion notch with a really nice flake that's taken off here. This is a percussion pit. These are some cut marks, some more cut marks here. These fossils are really nicely preserved. These bones are almost 2 million years old, but the cut marks actually look like they could have been made yesterday. The marks are still sharp right inside the cut. So this is the tooth mark distribution on the tibia or the shin bone of the antelope from Canberra and from FLK zinj. And then I'm comparing them with my different experimental models. So this is my carnivore-only model here. This is one of Blumenstein's, a model based on Blumenstein's experiments, the hammerstone to carnivore experiments. So these are where humans first butchered bones and then broke them for marrow, and then they threw them to the hyenas. So at Canberra, if we're looking at the distribution of tooth marks at Canberra, they're pretty randomly distributed. They don't show a clustering pattern, and so I'm showing the individual pits here. The tooth mark frequencies are pretty low at Canberra, and so this probably indicates carnivores were not very active at the site. On the tibia from FLK zinj, the tooth marks do occur in significantly dense clusters, and they resemble the hammerstone to carnivore model here more closely. So this pattern also looks pretty much the same for all the other bones of the skeleton at FLK zinj. So it looks like hominins had first access to carcasses, and carnivores were scavenging at FLK zinj. So looking at the frequency of tooth-marked bones in the different experimental assemblages, you can see that in the carnivore-only assemblages, these ones in red here, they all have a pretty high percentage of tooth-marked bone. And then in the green, these are the hammerstone to carnivore experiments. They have a significantly lower frequency of tooth-marked bone. And this makes sense there because there isn't much meat left on these bones to attract carnivores, so they're not chewing them as much, right? When we plot on the tooth mark frequency from Canberra, Canberra falls within or even slightly below the range of these hominin first assemblages. So each dot here represents a different analyst. We had 3 researchers doing the analysis, and then we have 3 levels in the excavation that we're looking at, so KS1, 2, and 3. So it seems like there's consistency between analysts in our study, which is good, and also consistency in our excavation through time. So there's this consistent pattern through time, suggesting that hominins were getting access to carcasses before carnivores were. And we can also look at the distribution of cut marks. This is the distribution of cut marks and percussion marking on the antelope femora from Canberra. And I'm analyzing the cut marks here using a model called the hot zone approach. And this helps to determine if cut marks were made before or after lions had access. So it helps to get at hunting versus scavenging by hominins. Now, it's been observed in feeding experiments that lions consume carcasses in this patterned sequence. and they don't always consume all of the flesh on bones. So given this, cut marks seen on anatomical areas where flesh never survives lion consumption should indicate primary access by hominins. This has been called the hot zone approach. It's an approach that was developed by Dominguez-Rodrigo and colleagues. I'm showing you here photos of the surviving flesh on limbs from my own lion feeding experiments. And this shows this, this similar pattern where lions typically consume flesh from the upper limb portions, but not the lower portions. So you can see that in both the forelimb and the hind limb, the upper part of the limb where there's more meat, lions tend to consume that fully, but they leave the bottom part of the limb pretty much untouched. So here are Dominguez-Rodrigo's cold zones mapped over the Canberra data. So these cold zones are opposite the hot zones. They're areas where flesh remains after felid consumption. And you can see here that the cut marks occur in the hot zones. So this suggests that hominins had primary access because there would have been no flesh left to remove in those areas if hominins were scavenging from lions. So this is just a bit about the cut mark evidence. So to summarize the interpretation of Canberra and Zinj based on my analysis, the cut mark evidence suggests that hominins had early access at least to small antelope. There are cut marks in the hot zones. There are low tooth mark frequencies at both sites, and the random distribution of tooth marks suggests that there wasn't much carnivore involvement, so probably carnivores had secondary access. So how were hominins gaining early access to these carcasses with their pretty basic Oldowan tool technology? They may have been hunting the smaller animals and aggressively scavenging the larger animals, so scaring off the carnivores before they were finished to gain access to the carcasses while there was still meat remaining. And if hunting for small animals is the case, Canberra may be the oldest evidence for hunting in the archaeological record that we have to date. There's some important implications here for understanding the evolution of human behavior. We know that hominins were foraging in a variety of habitats, from the grasslands at Canberra to the woodlands at FLK Zinj. And they were using stone tools to process both plant and animal foods. And the use of tools signals a behavioral shift away from a more ape-like subsistence strategy toward using culture to acquire food. Hominins were probably hunting small animals and scavenging larger ones. Now, hunting is a cooperative behavior, so hominins were almost certainly cooperating in this activity. And because hominins were getting early access to carcasses, they were getting meat in large packets that could have been shared. Now, large-scale food sharing is a behavior that we only see in humans, and so we might be seeing the origin of this behavior with the addition of meat to the diet. And then finally, Canberra and Zinj provide evidence that some sites were visited repeatedly over tens to hundreds of years by hominins, and these sites were the focus of butchery activities. Hominins left hundreds of stone tools and bones at these places. So it's likely that these favored places represent something like a home base where hominins would have returned to on a daily basis and shared food with extended family members. And this is something that we really only see in humans. So some key transitions in human evolution that we're seeing here are using culture to acquire food and cooperation and food sharing. So to end with, I just want to talk a little bit about some of my ongoing research. My work at Canberra was done through the Homa Peninsula Paleo Project, which is a collaborative project directed by Tom Plummer of the City University of New York. He was my PhD advisor. And then a team of researchers from different institutions, including Liverpool John Moores University in the UK and the Smithsonian. We also work closely with the National Museum of Kenya, which is where our expert field excavators are based. Here we are in excavation 1. These guys are from the National Museum of Kenya. So I mentioned that we started exploring new sites in the area around Canberra. And a couple years ago, or last year, we started an excavation at a new site called Nyayanga right here. And it's just a few kilometers away from Canberra. This is what it looks like at Nyayanga with Lake Victoria in the background. It's a nice view from that site. So the funny thing about the Homa Peninsula and about Canberra is that we found literally thousands of Oldowan stone tools and bones that were clearly butchered by hominins, but until a couple years ago, we actually had not found any hominin fossils. A couple years ago, while we were surveying at Nyanga, we were actually thrilled to find this tooth. This is a tooth of Paranthropus boisei. It's the species that has those really huge, easily identifiable molars. This is the skull that the Leakeys found at Olduvai. This is the tooth that we found, and it's clearly from this species Paranthropus. So we were pretty excited when we found that tooth, and we decided to put in a full excavation in 2016 after we found that. We actually found the tooth under this tree here. This was our lunch spot because it's the only shade at Nyanga, and someone happened to pick it up while we were sitting around during lunch one day. So we put in this big excavation at Nyanga in 2016, and all these guys here that are helping us out are either trained excavators from the National Museum of Kenya, and some of them are local people from the surrounding area. We try to make sure that we have a good relationship with the surrounding community by creating some jobs while we're there. And what we found in the excavation was not a Paranthropus, unfortunately, but we did find a hippo skeleton. And this might just look like a pile of bones to you, but this is, it looks like we have the whole hippo skeleton here. Here's some of the close-ups of what we found. This is the tooth from that hippo. And one of the things that had us really excited about the excavation is that we found some stone tools right in with the hippo skeleton. We found a hammerstone, and we found 3 flakes. This is one of them. And the flakes were basically embedded right in with the skeleton. It's just been excavated. We haven't had a chance to look at it yet to see if there are cut marks on the bones. The fossils are now in the National Museum of Kenya, and we'll be returning next field season to analyze them further. But I'll be surprised if there are not cut marks on these fossils, given that the stone tools are mixed right in with them. We've actually found a lot of bone on the surface of the excavation, so those fossils are not in context, but some of them, like this one, do actually have cut marks on them. You can see those marks here and here. So our geologists are working to try to get a good date for Nyanga, but based on the fauna, it looks like it might be a little bit older than Canberra. It might be about 2.4 million years old. And if this is the case, this is some of the oldest clearly cut marked bone in the archaeological record. So I'm looking forward to the things that we're potentially going to learn about hominin subsistence and behavior at these sites. So these kinds of projects are never done in isolation, so I just want to thank these people and these institutions that I collaborated with and who provided me with funding, and thank you all for coming and listening.